I'm Dr. Brad Dengler, the lead author of this study entitled Quantitative Pupillometry Predicts Return to Play and Tracks the Clinical Evolution of Mild Traumatic Brain Injury in United States Military Academy Cadets, a Military Traumatic Brain Injury Initiative study. I'm currently a neurosurgeon assigned to Walter Reed and the director of the Military Traumatic Brain Injury Initiative, a research center at the Uniformed Services University of the Health Sciences. Assessing and diagnosing a mild traumatic brain injury is a difficult process because there is currently no standard objective measurement for doctors to use. The most comprehensive assessment tool that currently exists is the Sports Concussion Assessment Tool 6 version. It is difficult to use because both time-consuming and requires training prior to its implementation, which limits the amount of people who can use it, and also how many times it can be used before running out of time. Identifying a faster, more accurate, and more objective assessment tool that doesn't require extensive training would lead to faster and more appropriate diagnoses and treatments. Our objective in this study was to determine the utility of the pupillary light reflex as a biomarker of mild traumatic brain injury. This study is the first to evaluate that rapid objective measure for mild traumatic brain injury and link it to the length of time until return to duty. If successful, it would be easier to use this test by medics and sideline personnel at sporting events. Our study was conducted at the United States Military Academy. Upon enrollment, 1,300 cadets underwent a standard battery of tests, including the balance error score, the Sports Concussion Assessment Tool 5th edition, and the standard assessment of concussion. Pupillary responses were recorded using the Neuroptics PLR 3000 pupillometer at all time points. During the study, 68 cadets sustained a mild traumatic brain injury, which was mainly a result of training events or sports-related concussions. They were evaluated using the same battery of tests at the baseline time point before injury occurred, within 48 hours of the injury, and at the initiation of the graded return to activity protocol and an unrestricted return to activity. The pupillary responses post-mild traumatic brain injury demonstrated a pattern of significant change that returned to normal for several measures, which is the two graphs located at the bottom of the slide. And those two measures were the difference between the constricted initial pupillary diameter, dilation velocity, and percent of pupillary constriction. While not all of the pupillary measures followed the clinical parameters, several did. The two clinical parameters illustrated above are the Sports Concussion Assessment Tool, along with the balance error score, which both increased and then returned to baseline by the time the cadets went to unrestricted return to play. The combination of dilation velocity and maximum constriction velocity demonstrates a moderate ability with an AUC of 0.71 to predict who can return to duty before or after 21 days of treatment, as seen here in the receiver operating characteristic curves for those two pupillometry variables. This study's findings indicate that quantitative pupillometry has the potential to assist with injury identification and prediction of symptom severity and duration when administered after a mild traumatic brain injury.